Two steps forwards, run 6 paces. Tell me where you are.
On this lecture and the anatomical lecture, the learning objectives are to list and describe the functions of the kidneys, describe the gross and microscopic anatomy of the kidneys, list the main blood vessels associated with the kidneys. It is all about the kidneys, apparently. Define the terms glomerular filtration, renal threshold, polyuria, polydipsia, urolithiasis, and uremia. Describe the production of urine. Describe the mechanisms that affect urine volume. Describe the structures and functions of the ureters and bladder. Describe the processes involved in micturition. And describe the structure and functions of the urethra. So the urinary system, as we know, contains two kidneys, two ureters, a urinary bladder, and a urethra. A lot of animals can live with one kidney, although not all animals. So that's a little bit species dependent and sort of dependency upon those having both kidneys. Just going back here, it's important to note that the right kidney in most mammals, with some variation, but in most mammals, the right kidney sits cranial in the body, more so than the left kidney. So the function of the urinary system is to regulate the volume and composition of body fluids and excrete unwanted material. As products enter the urinary system, some are kept and recycled, others are tossed out into the urine as waste products. Carbon dioxide, so waste products found in urine, we've got carbon dioxide and water from carbohydrate and fat metabolism. A lot of it is nitrogenous wastes, such as urea, which is a predominant nitrogenous waste. It's the result of breakdown and metabolism of protein. Bile salts and pigments from red blood cell breakdown, and various salts from tissue breakdown and excessive consumption. The body has several routes by which waste products are eliminated. So other than the urinary tract system, we've got the respiratory system, the sweat glands, the digestive system, and then of course the urinary system that we're talking about. So not all the wastes are eliminated through the urinary tract. Some of them are, and those that are we'll talk about in a little bit of detail as we go through. So the urinary system is the single most important route of waste product removal in the body. So we have other methods, sweat glands, respiratory tract, digestive, but the urinary tract has an important role in getting rid of the majority of the toxins that would otherwise build up in the bloodstream. So imagine what would happen if these waste products weren't allowed to actually escape the body, so if they maintain themselves in the bloodstream. Or imagine if the body excreted products that it actually needed to recycle, what would happen? In the cases where the nitrogenous waste and other waste would hang around in the bloodstream, the animals actually become toxic. So their blood becomes normally quite acidic, depending on what's being held back. But they can get problems with acidosis, and of course they start to vomit, they start to not feel good, they become nauseous because they're literally becoming toxic from the inside out. And then on the other side, if the body's excreting products that it actually needs to recycle, like protein especially, then we start getting all sorts of other issues associated with a lack of nutrients and a lack of some of these specific chemicals that are required for regular body functioning. The urinary tract removes nearly all soluble waste from the blood and it transports it out of the body, gets it out of the body. The urinary system is also a major route for the elimination of excess water from the body so that the body doesn't hang on to too much water as it's working away. So we're going to take a closer look, bring in the kidney. It's located in the dorsal abdominal area, ventral to and on either side of the first few lumbar vertebrae. So essentially it's right close to the spine and it's on either side of the first few lumbar vertebrae. Okay, so it's really far up or dorsal in the animal. It's retroperitoneal to the abdominal cavity, so that means that it actually doesn't sit within the abdominal cavity. It sits between the peritoneum, which is the abdominal cavity membrane, and the dorsal abdominal muscles, the epaxial muscles essentially. So keeping in mind that when we enter into the abdomen, we're not into the area that we can view and work on the kidneys. Okay, they sit just past the peritoneum. In most domestic species, as I mentioned before, the right kidney is a little more cranial than the left, and there's a thick layer of perirenal fat that commonly surrounds kidneys to help protect them from pressure exerted by surrounding organs. It also, of course, plays a role in thermoregulation because they tend to have an abundance of fat around the renal vein and the renal artery. So based on the anatomy lecture, we know that the kidney produces urine and filters blood. So of course, altogether, this helps to maintain homeostasis. The kidneys work on sort of a one in, one out principle for electrolyte and waste removal, which we'll talk about in a second. So the homeostasis maintenance is in part contributed by the kidneys, of course, through manipulating the composition of blood plasma. In this way, the kidney can regulate such things as body acid-base balance and fluid electrolyte balances, and the kidney has to filter enough water and electrolytes out of the blood to equal the amount that's being put in from other sources. So that's where the one in, one out comes in. So if the other areas of the body are breaking down protein and they're contributing substrates or these little molecules into the kidney to be passed through, the kidney has to decide what stays in and what stays out. But in order to maintain homeostasis, it's that continuous one in, one out mechanism. So example, the levels of sodium, potassium chloride, and nitrogenous wastes, which typically we talk about as urea, in plasma, so in the blood plasma, they have to be maintained within specific narrow concentration limits for health and life to continue. If the kidney fails to remove these substances from plasma adequately, their concentrations can rise to really toxic levels, and that's what I was talking about before. So this leads to illness and potentially even death, especially if they're left to be uncontrolled. Kidney functions, we have a lot. We've got blood filtration, reabsorption and secretion, fluid balance regulation, acid balance regulation, hormone production, and blood pressure regulation. So blood filtration, reabsorption, and secretion, essentially the blood is filtered, useful substances are returned to circulation, and the waste products are secreted from the bloodstream to the fluid that eventually becomes the urine. We'll talk a lot about this process. Fluid balance regulation, the amount of urine produced depends on the amount of water it contains, which helps ensure that the body contains the right amount of water to maintain a healthy internal environment. If the body has excess water and needs to get rid of it, more urine is formed, and that's called diuresis. If the body needs to conserve water, less urine will be produced, and the animal will pass very little urine. That's called oliguria, or no urine at all, which is called anuria. The amount of water contained in the urine is under control of the the antidiuretic hormone and aldosterone, and we'll talk about those a little bit later on as well. Acid-base balance regulation, the kidneys help maintain acid-base homeostasis by their ability to remove acidic hydrogen and basic bicarbonate ions from the blood and excrete them in the urine. By eliminating these ions in the appropriate amounts, blood pH can be maintained in the proper range. Hormone production, the kidneys have close associations with the endocrine system, the hormones that keep that keep everything functioning in the body. So that's the thyroid system, the endocrine system. The kidneys produce hormones, they regulate the release of hormones from other organs, and they are themselves influenced by hormones. So example, the kidney can influence the rate of release of the antidiuretic hormone from the posterior pituitary gland, and aldosterone, the mineral corticoid that's secreted by the cortex of the adrenal gland. The kidneys are also influenced by both of these hormones, so they can tell those hormones how much to secrete or tell those glands how much to secrete in the way of hormones, and then they're also under control of those hormones as well. Specialized cells in the kidneys produce erythropoietin, and that's the hormone that's necessary for red blood cell production and for some prostaglandins. So we'll talk about a little bit of those later on, especially when we talk about kidney failure as a case study. So then blood pressure regulation, the kidneys contain internal receptors that monitor blood pressure. When the blood pressure falls, the kidneys secrete a hormone called renin. The release of renin will start a cascade of reactions that will result in vasoconstriction and retention of sodium and water. By increasing the fluid volume of the blood pressure also is increased. And we'll talk more about all of that as we go on. So quick anatomical review. with the kidney. So looking at the kidney, we've got a real life kidney and then of course a cross section of the kidney. So we have on the outermost layer through here, we have essentially the renal capsule. And then we work our way, the next layer is the cortex of the kidney. So that's sort of the next superficial layer. Beyond the cortex, we have the medulla, which is sort of that center layer. In the medulla, we have the medullary pyramids, or otherwise known as the renal pyramids, through here. So the medulla is essentially the central layer, or a thick inner layer of the kidney. So you've got all those renal pyramids, which then connect here at the renal papilla, and then they enter the byproducts into the minor calyces. The minor calyces then empty into the major calyces, which are the two larger branches, and then, of course, eventually into the renal pelvis. The renal pelvis then becomes the ureter. The hilus, or the hilum, whichever you choose to call it, that's the indent of the kidney, and that's where the lymphatic vessels enter, the renal artery enters, and the renal vein. It's important to know, if we're talking about cows are multilobulated, so they're lumpy on the outside. I haven't been able to find any information that substantiates exactly why this is. Some people thought maybe it's to increase surface area, but then when you perform a cross-section on cow kidneys, they look the same on the inside. So they still have the same medulla, and they have that pyramid type formation as well. So it's argued that that's not necessarily correct, that it's for surface area. Some people note that it's vestigial, so it's a remnant of evolution. But again, I would love to know if you find out why cows are lobulated. So then looking at the cross section, this is exactly what it looks like. So we've got the capsule on the outside. The cortex is sort of the first innermost layer. And then beyond the cortex, we have the medulla, which includes all these renal pyramids right through here. In between the renal pyramids are the renal columns. And then the renal papilla, which you can't really see overly well, but connects to the minor calyces and then the major calyces into the renal pelvis, as noted here, and then of course out the ureter. So that's a very quick review. Okay, so then we're going to look at sort of the more important part, which is the microscopic anatomy of the kidneys. So within the cortex and the medulla of the kidney, they're packed with hundreds of thousands of microscopic filtering, reabsorbing, and secreting units called the nephrons. The nephron is the basic functioning unit of the kidney, and it's the smallest part of the kidney that can actually carry out its functions, right? So that's what it means, smallest basic functioning component. Each nephron is composed of a renal corpuscle, a proximal convoluted tubule, a loop of Henle, and a distal convoluted tubule. And we'll go through each of those and their functions in a fair bit of detail. So numbers matter. I mentioned that they have hundreds of thousands of these nephrons in their kidney. Every species has a different number of nephrons depending on their needs. Medium-sized cat has 200,000, roughly. Medium-sized dog has roughly 700,000. Sheep, pigs, humans, up to 1 million. Cattle, like 4 million. So what does it affect? It depends, and it's arguable what it affects specifically. So some say that this can impact the way that the animals actually metabolize certain drugs, especially drugs that are metabolized primarily through the kidneys. So when we look at cats are really sensitive to a lot of drugs that are metabolized through the kidneys, like ASA, so aspirin. Drugs like that can really just shut down their kidneys. Likewise, things like lily toxicity in certain flowers and plants, it can really just shut down cat kidneys. So one school of thought that specialists identify with is that because of their lack of nephrons, because they have a lower end on the spectrum of the number of nephrons, they have a lower ability to actually process these nephrons. But at the same time, we have to keep in mind that it's not just the kidneys that are involved with metabolism, that there's differences in the number of enzymes that they produce as well, which help break down these drugs. So it's just a thought. Cats are very sensitive to things like acetaminophen, ASA, like I mentioned, etc. And overall, they just metabolize them very slowly and sometimes very ineffectively to the point where they overload the kidneys. So we're going to take a closer look. So the renal corpuscle, right here, this area, it includes the capsular space Bowman's capsule, and the glomerulus. The function is to filter blood in the first stage of urine production. And then we move on to the proximal convoluted tubule. So it's the first tubule, and of course convoluted means that it's nice and windy. So here's the proximal convoluted tubule. Please ignore the cat snoring in the background. It's lined with cuboidal epithelial cells with a brush border. So the purpose in this case of the brush border, essentially they're little microvilli on the cell wall, or not the cell wall, on the cell membrane, and they extend the surface area. So they increase the surface area to assist with absorption and secretion. So looking at this, let's see here, the renal corpuscle, as I mentioned before, it's composed of the glomerulus surrounded by the Bowman's capsule, and it's located in the cortex of the kidney. It's made up of the glomerulus and the Bowman's capsule, as mentioned about 5 times by now. The glomerulus is a tuft of glomerular capillaries, and the Bowman's capsule is a double-walled capsule that surrounds the glomerulus. The function of the renal corpuscle is to filter blood in the first stage of urine production. The fluid that's filtered out of this portion of the nephron is called the glomerular filtrate. The proximal convoluted tubule is a continuation of the capsular space of the Bowman's capsule. So it's kind of like this continuation of a very thin membrane. It's the longest part of the tubular system of the nephron, and the epithelial cells that line the PCT are cuboidal, and they have that brush border like I mentioned. So that is really, really important, and it increases the surface area by a factor of 20. So it's pretty crazy. It's especially important in the whole reabsorption process that happens, which we'll talk about in detail. And that's about it for the PCT and the renal corpuscle. So just a little bit of a refresher. This sits within both the cortex of the kidney, and then when we talk about the loop of Henle, it reaches down into the medulla of the kidney. Okay, so talking about the PCT and the renal corpuscle sits within the cortex, this layer here. So looking a closer look at the PCT brush border, so this is a typical little epithelial cell with microvilli, and again increases that surface area by a factor of 20, which is crazy. All right, then we've got the loop of Henle. So the loop of Henle continues from the PCT and it descends down into the medulla of the kidney, as I mentioned before. Then it makes a U-turn and heads back up to the cortex. The descending part of the loop of Henle has cuboidal epithelial cells, similar to the cells of PCT, including the brush border. As the loop of Henle makes its U-turn, the walls become thinner and the epithelial cells flatten into simple squamous epithelial cells, and they actually lose that brush border. The lumen gets narrower here too. As the loop of Henle ascends back into the cortex, its walls become thicker again, but the cells don't regain the brush border. They don't really need it at this stage. The lumen also expands as the loop of Henle ascends to the cortex. And then we have the distal convoluted tubule. So this follows a twisting path through the cortex. Even though it's called the convoluted tubule, it's not as twisted as the PCT. The distal convoluted tubules from all the nephrons in the kidney empty into a series of tubules called the collecting ducts. So that's right here. So each time a nephron connects and dumps into this channel, which eventually is dumping that urine type product further into the kidney, they are all attached to the collecting duct. Okay, so it's like a little series of drains. The collecting ducts carry tubular filtrate through the medulla into the calyces, which lead to the renal pelvis. The collecting ducts also play an important role in urine volume because they're the primary site of action of the antidiuretic hormone, which we'll talk about later. Potassium regulation and acid-base balance control are two other important functions that take place in the collecting ducts. So then once they get all the substrates, the byproducts which become urine, they dump it into the collecting ducts. It goes on through the minor calyces to the major calyces and out to the renal pelvis, which eventually becomes the ureter. So nerve supply to the urinary tract system overall, so to the kidneys to produce urine, and then on through the ureters and the bladder. Mainly it's under control of the sympathetic nervous system. So that's important to know because we know that that's stimulated with the fight or flight reflex. So during fight or flight, urine production is shut down. So we have blood flow. Blood flow blood flow through the kidney, going back to the kidney overall, it's the most extensive capillary network of any organ in the body. Every 4 to 5 minutes, the body's entire blood volume filters through the kidneys, which is pretty amazing. 25% of the blood pumped by the heart gets pumped directly to the kidneys. And I've posted a video that explains this really well. It should be in the playlist for anatomy and physiology. We're going to look at blood flow through the kidney. So looking at the gross anatomy, don't forget that the aorta runs down the length of the abdomen, and it kind of sits along the spine almost, right at the base, right at the back of the abdomen. So the aorta branches off and provides oxygenated blood to the kidneys. It becomes the renal arteries. And of course, the blue there is indicated that's the caudal vena cava, and it branches off to become the renal veins. So looking at the blood flow through the kidneys, so the renal artery branches off the abdominal portion of the aorta. It enters the kidney at the hilus, which is the indented portion of the kidney. It then divides and subdivides into smaller arteries and arterioles until it becomes a series of afferent glomerular arterioles. The afferent glomerular arterioles carry blood into the glomerular capillaries of the renal corpuscle, and the glomerular capillaries are a continuation of the afferent arterioles. They filter some of the plasma out of the blood, which enters the capsular space of the Bowman's capsule, at which point it's known as glomerular filtrate. Not all of the plasma is filtered out. If it were, the blood flow would come to a screeching halt. The blood in the glomerular capillaries leaves the glomerulus and enters the efferent glomerular arterioles. Notice that the blood is still arterial blood at this point because oxygen exchange hasn't taken place yet. This is the only place in the body where blood entering and leaving the capillaries is oxygenated blood. Okay, a little bit different. The efferent glomerular arterioles divide into a network of capillaries that surround the rest of the nephron, and these capillaries are known as the peritubular capillaries. We should have potentially a photo of those coming up. Oxygen transfer to the cells of the nephron takes place here. At this level, substances are taken out of the tubular filtrate and put back into the blood. So this is known as tubular reabsorption. Other substances are secreted from the blood into the tubules at this level. This is called tubular secretion. We'll talk about these a lot more as we go on. So here's, actually I'll carry on here. The pretubular capillaries that surround the nephron converge to form venules that in turn converge to form larger veins and eventually become the renal vein. And then the renal vein takes all that deoxygenated blood out of the kidney at the hilus, essentially it enters and it joins the abdominal portion of the caudal vena cava. So with regard to waste content, the blood in the renal veins is the purest blood in the body, okay, because all of the waste products have been removed. So keep that in mind. The blood in the renal vein is the happiest toxin-free blood in the body. So here's a little pictorial, again, of how this is working. So you've got the renal artery bringing oxygenated blood that branches into the interlobar arteries. The arcuate arteries then are branches off of the interlobar arteries. And then on a very smaller framework, the interlobular arteries are bringing blood up toward the glomerulus. And then it branches off further into the afferent glomerular arterioles. And then, of course, the glomerular capillaries. Say that 500 times fast. So now the nephron, or specifically the glomerulus, is oxygenated. Okay, so it's happy, it's oxygenated. So then we have to bring that deoxygenated blood back. The efferent glomerular arterioles exchange and sort of branch into the peritubular capillaries, the interlobular veins, the arcuate veins, the interlobar veins, and then branching off into the renal vein. At this point, the blood has left the kidney. Okay. Filtration of blood. So this occurs in the corpuscle. Normally, capillaries are found between so I'll talk about the specialness. So glomerular capillaries are very special. They're under really high pressure. So they're under this about 30% of the pressure of the aorta, which is pretty amazing considering the aorta is the highest pressure in the body. And they are between two arterioles, which we'll talk about here in a sec. So they are abnormal in that way. Normally, capillaries are found between arterioles and venules. They connect the two, and they have really low blood pressure. So they sort of pretty much are the opposite to that. So the high blood pressure in these capillaries is created by a difference in the size between the afferent and efferent glomerular arterioles. The diameter of the efferent glomerular arteriole is smaller than that of the afferent arteriole. So this causes the blood to build up within the glomerular capillaries. The high blood pressure in the glomerular capillaries forces some of the plasma out of the capillaries and into the capsular space of the Bowman's capsule. The transfer of plasma out of the glomerular capillaries is helped by the presence of many fenestrations, so little holes or pores, in the capillary endothelium. These fenestrations are larger than the fenestrations found in the endothelium of other capillaries, and they actually allow more fluid to leave the bloodstream. This fluid is known now as the glomerular filtrate when it enters the capsular space, and it's similar to plasma, except that it virtually contains no proteins, or it should contain no proteins. The filtration of blood in the glomerulus is mainly a size-regulated process. So molecules within the blood that fit through the fenestration will move through the plasma and up as part of the glomerular filtrate. Larger molecules that can't fit through the fenestrations will stay in the bloodstream. Most of the plasma protein molecules are too big to pass through those little fenestrations. So blood cells are also too big to pass through the fenestrations, and if the endothelium of the glomerulus is damaged, it's really important to note that protein and blood cells can actually leak into the glomerular filtrate. So because there's no mechanisms to get the proteins back into the bloodstream through tubular reabsorption, they show up as abnormal substrates or constituents of urine. The presence of abnormal amounts of proteins in urine can be used as an indicator of glomerular damage. And we'll talk about that a lot later because we'll talk about renal disease and sort of the clinical signs and then the blood chemistries that show up as well as the urinalysis results. So wastes are removed. That's the goal of the filtration of blood. And we talked about nitrogenous wastes such as creatinine and urea. When these wastes aren't removed, so when there's a buildup of these wastes in the bloodstream, when it's a buildup of creatinine, it's called azotemia. And when it's a buildup of urea, it's called uremia. And we'll talk about that a little bit later when it comes to talking about renal disease. The filtration of blood in the glomerulus is mainly a size-regulated process. Molecules within the blood that fit through the fenestrations will move through with the plasma and end up as part of the glomerular filtrate. Larger molecules that can't fit, they'll stay in the bloodstream. Most of the plasma proteins are too large. Blood cells are too large, as I mentioned before. And then, of course, if there's damage to them, they can leak. So those are the key points to bring home. High blood pressure, again, is responsible for this, helps push the plasma out and pushes it through that filter. And then glomerular filtration rate, or GFR, is how fast the plasma is actually filtered through the glomerulus. So proteinuria is a term given to protein in the urine. The glomerular membrane is allowing larger proteins through the barrier rather than hanging onto them to maintain in the bloodstream. So that's what's happening to create proteinuria. The two major groups of serum proteins in the blood are called albumin and globulins. Albumin is abundant in the blood, and it accounts for about 50% of all serum proteins. It has important functions that include pulling water into the capillaries and maintaining the right amount of water in the circulatory systems, as well as binding and carrying substances that are poorly soluble in water. So you can imagine if the body's pumping out albumin into the urine and getting rid of it, we start to get what's called a protein losing disease. So lots of different reasons that this can happen. So oftentimes we will perform urinalyses on an animal. So we're checking the color, the odor, the clarity of the urine, and then we're performing a chemical analysis called a dipstick. And it's on the dipstick that we get to identify whether or not there's protein in the urine. So if we're seeing proteinuria, protein in the urine on the dipstick, that's an indicator that another disease process might be happening. Commonly, we see proteinuria in renal disease, especially chronic renal disease, glomerular disease, such as glomerulonephritis, so inflammation of the nephrons of the kidney, and amyloidosis. Secondary pyelonephritis, which is inflammation of the kidney, or the nephrons specifically. Leptospirosis, which is a disease that's passed through contaminated water from wildlife. Renal neoplasia, and nephroliths. And of course, nephroliths. It's hard to say. Those are kidney stones, which are not ideal in any case. Then we can get transient proteinuria, so proteinuria that comes and then goes fairly quickly, and that's often related to sort of temporary conditions such as heat, stress, seizures, venous congestion, fever, and extreme exercise can all cause transient proteinuria. Like I mentioned before, we detect it during a urinalysis. This is a dipstick. So we literally dip it in the urine, let it sit for a period of time, and then we compare it to its guide, and it tells us how much protein's in the urine. Persistent proteinuria could be a flag for renal disease. Just to keep that in mind, we'll talk about renal disease later. By the time the animal is actually showing clinical signs such as polyuria, polydipsia, lack of muscle tone, etc., that's normally at the point which 2/3 of the nephrons are destroyed. Into the process of reabsorption within the nephrons. So both good things and bad things sneak out of these little glomerular capillaries. Substances to be reabsorbed pass out of the tubular lumen through or between tubular epithelial cells, and the substances to be reabsorbed then enter the interstitial fluid and pass through the endothelium back into the peritubular capillaries. So these substances, as soon as they pass out of circulation and into the capsular space, they're considered out of the body. So they're just considered in a piece, an area of the body that's not really considered in the body. It's kind of weird. They contain bad things quite often, such as nitrogenous waste, bile salts, etc., and then some good things like sodium, potassium, calcium, etc. So generally, the glomerular filtrate is composed of fluid containing the small molecules that fit through the glomerular fenestrations. Some of them are waste products, like I mentioned, some of them are good things. So, because it needs to maintain homeostasis, the body, it needs to undergo reabsorption. So if it's losing too many of the good things, then we have to go through reabsorption. Some of the most important of these substances that we reabsorb in the body are sodium, potassium, calcium, magnesium, glucose, amino acids, chloride, bicarbonate, and water. So there's a lot of them. So there's a mechanism in place to get these useful substances back into the body through the bloodstream. And of course, that mechanism is reabsorption, which we'll talk about in detail now. So the substances, they've gone through the glomerulus, they're escaping through the capillaries, and now they make their way through the proximal convoluted tubule. This is where reabsorption really begins, and the majority of reabsorption takes place. At which time once it's entered into the proximal convoluted tubule, it's called tubular filtrate. So looking at sodium reabsorption, the goal here is to pump sodium out of the filtrate and back into the blood, right? It's a good thing, we want to keep it. So sodium starts off essentially, so I'll go through the technical terminology. So as the glomerular filtrate enters the lumen of the PCT, the sodium is actively pumped out of the fluid back into the bloodstream. So basically it starts off by floating in the nothingness of the PCT internal space. So it's just hanging out in the proximal convoluted tubule. Then it has to make its way back sort of into that body proper, so actually into the body. So it has to latch on to a carrier protein in order to get past the doors and actually into the PCT epithelial cells, because the PCT epithelial cells are the ticket to getting transferred back into that whole body proper space. So once it's into the PCT epithelial cells, it has to be actively pumped out of the cell, which requires energy, into the interstitial space, so the space between cells, where it can get picked up into the bloodstream through the peritubular capillaries. So essentially it has to act as a hitchhiker with a carrier protein to get back into the PCT epithelial cells, where it then gets pumped into the interstitial space and then picked up by the capillaries. Pretty sweet. Sodium ions are also reabsorbed in the ascending part of the loop of Henle and the DCT, where they are usually exchanged for hydrogen, ammonium, or potassium ions that are then secreted into the tubular filtrate. So they pick up the sodium and get rid of hydrogen, ammonium, or potassium ions. This exchange process is under the influence of aldosterone, which is the mineralocorticoid hormone produced in the cortex of the adrenal gland. Okay, it all goes back to the endocrine glands. Potassium diffuses out of the tubular filtrate by moving between the epithelial cells into the interstitial space, and then, of course, into those peritubular capillaries. The potassium's reabsorption takes place in the PCT, the ascending part of the loop of Henle, and the DCT. Magnesium is reabsorbed from the PCT, the ascending loop of Henle, and the collecting duct. The parathyroid hormone release increases the reabsorption of magnesium. Potassium and calcium reabsorption takes place in the PCT, so the ascending loop of Henle and the distal convoluted tubule. The calcium moves from the filtrate under the influence of vitamin D, under the influence of the parathyroid hormone, and calcitonin. Okay, so it's all, calcium does not act on its own. It's totally directed by other forces. So the interesting thing here, just a side note, interesting fact, how do humans acquire vitamin D? We know it as the sunshine vitamin. That's very important. But what's really neat about vitamin D is it's not really a vitamin, technically. It's actually more like a hormone. See, the importance of this is that it helps regulate the mineral metabolism, such as calcium, of course, and subsequently it maintains calcium balance within the actual bones themselves. So it's thought that this vitamin, vitamin D, plays a role in affecting the expression of over 200 genes in the body as well, which is pretty crazy. The oil that naturally comes from our skin is a type of cholesterol. Okay, so this is how vitamin D is synthesized. So it starts off as an oil that comes from our skin. When we stand in the direct sunlight, the ultraviolet rays react with this oil, and they actually radiate the oil. The radiation breaks some chemical bonds in the oil so that it forms a different molecule. It is then absorbed back into the skin and enters the bloodstream. So that is vitamin D. So once those cholesterol molecules are broken down with ultraviolet radiation, they become vitamin D, which is then reabsorbed through our skin back into our bloodstream. Isn't that crazy? I find that really neat. We all call it the sunshine vitamin, but then we don't actually know why. Cats, birds, anything with fur, the vitamin D can actually get lost on their fur or their feathers. So then it doesn't as easily get absorbed into the skin and therefore into the bloodstream. So what they end up doing, which is super cool, is they end up grooming to assist with that. So they groom, they lick off those broken down oils, and then it gets absorbed into their body that way. So it's kind of neat. So when you see cats and dogs, especially cats, laying in the sunshine, picking that really sunny spot in your house and grooming like crazy, that's actually a natural mechanism to acquire more vitamin D. Kind of neat, interesting fact. All right, so reabsorption, back to reabsorption. Chloride diffuses from the tubular filtrate into the epithelial cells and interstitial space in response to electrical imbalances created by the sodium removal. Remember that sodium and chloride sort of have that continuous back and forth effect to a point to maintain negative inside the cell. Some of the water in the filtrate moves into the interstitial space and pretubular capillaries by osmosis once sodium, glucose, amino acids, and chloride have left the tubular filtrate. So about 65% of all tubular reabsorption takes place in the PCT, where about 80% of the water, sodium, chloride, bicarbonate, and 100% of the glucose and amino acids in the tubular filtrate are reabsorbed. Additional reabsorption takes place in the loop of Henle, DCT, and the collecting ducts. Really important clinical application. So not everything can get reabsorbed. Only so much can get reabsorbed, and to a point, it's a one in, one out. So there's a reabsorption threshold. Here we'll talk about the clinical application of the renal threshold of glucose, the renal reabsorption threshold of glucose. So when the body is producing too much glucose in the bloodstream, such as during diabetes mellitus, when the animal doesn't have a good control of their insulin, they're not producing enough insulin, or they're not producing any insulin, what can end up happening, or what ends up happening, is that the glucose gets overproduced in their bloodstream. So when it works its way, the blood works its way down to the kidneys. Normally the kidneys would filter out and then reabsorb glucose as much as possible, but in the case of diabetes mellitus, high glucose maxes out the threshold, so then we get glucosuria. Glucosuria is when we start to see high levels of glucose in the urine. And then, of course, glucose, like many ions, will draw water with it. So we're getting a dumping of glucose in the urine on its way out. It doesn't go out alone, it's got to take out water with it. So the glucose actually hangs onto the water as well. So you get an excessive amount of urine production, which is polyuria, and then a compensatory action, so where they're compensating for the huge amount of urine being produced, they'll go through polydipsia. So just because of diabetes mellitus, again, we're seeing a lack of insulin production. It ends up having high levels of glucose in the blood. When the kidney tries to reabsorb that glucose, it just can't handle it, it's too much. We see glucose in the urine, and then of course, that urine with glucose in it is larger in volume because the glucose, as it's excreted, it hangs on to water molecules, okay? So then we get polyuria, polydipsia, and glucosuria. So one of our key tests for diabetes is to check more than one sample, so check sort of chronic occurrences of glucosuria. Oops, sorry. So going back now, we talk about secretion. So secretion primarily occurs in the DCT. Hydrogen, potassium, and ammonia are eliminated by secretion. So this is where it's spitting out some of those nitrogenous wastes. Some medications are also eliminated from the body by secretion, such as penicillin and sulfonamide antibiotics, which is really great if there's a bladder infection and they're sensitive to those antibiotics, because it will cause the antibiotics to actually have a local effect in the urine. So urine volume regulation is generally determined by amount of water contained in the tubular filtrate when it reaches the renal pelvis. It's overall controlled by the actions of the antidiuretic hormone and aldosterone. The antidiuretic hormone acts on the DCT and the collecting ducts to promote water reabsorption. Aldosterone increases reabsorption of sodium, and this causes the osmotic imbalance that encourages water to hang on to the sodium. So water, again, has to follow the sodium out of the tubular filtrate and into the blood. Where sodium goes, water will follow. Okay, so based on, it's very similar to the glucose drawing water out of the body into the urine. Very similar that where sodium will go, water will follow. So then we have this fun condition called diabetes insipidus. So the pituitary gland in this case is not releasing enough of the antidiuretic hormone. So let's break that down. Diuresis is the act of the body getting rid of water. So we take diuretics to get rid of water. So an antidiuretic hormone is preventing a loss of water. Okay, so it's a natural occurrence and it prevents an abnormal loss of water. Diabetes insipidus is this disease whereby the pituitary gland is not releasing enough antidiuretic hormone, which would, of course, result in diuresis, or the collecting duct is unable to actually respond to adequate amounts of the antidiuretic hormone. Either way, the body is undergoing diuresis. Lots of water is lost in the urine, so we're creating huge volumes of urine, so we've got polyuria, and lots of drinking, of course, is happening to compensate, so we've got polydipsia. It looks a lot like diabetes mellitus. So from afar, it looks a lot like diabetes mellitus. The difference is the balance of electrolytes will be different, will be changed. Okay, these dogs are continuously diuresing. And the glucose will be found in the urine for cats and dogs that have diabetes mellitus. Okay, we won't get glucose in the urine of diabetes insipidus. And in fact, insipid refers to tasteless. So back in the day when they were discovering all these medical problems, we would at one point taste the urine. I say we, but it's not my time. A long time ago. They would taste the urine and find that diabetes mellitus has sweet glucose flavored urine, right? Sweet urine, sugary urine. Whereas diabetes insipidus, the name comes from tasteless. All right, and then another role for the kidneys, because it's all about the kidneys. Blood pressure regulation. So of course, homeostasis is maintained through regulation of blood pressure in part. A decrease in blood pressure, when that occurs, renin-angiotensin-aldosterone system kicks in. So there are blood pressure sensors located in the afferent glomerular arterioles, and those are called the juxtaglomerular cells. And there are sodium sensors located in the ascending loop of Henle, the macula densa. Low blood pressure or low sodium creates this release of renin. Renin is an enzyme that facilitates the splitting of angiotensin 1 from angiotensin. And then we'll talk about that in a second here. Well, and then the angiotensin 1 is then converted to angiotensin 2 by the angiotensin converting enzyme. So we'll talk about that there. So renin is released from the body because of a drop in blood pressure or a drop in sodium. And then, of course, the renin splits the angiotensin, and then it's converted into angiotensin 2 through the angiotensin converting enzyme. Angiotensin 2 directly stimulates the arterial constriction and release of aldosterone. Okay, so it's immediately stimulating vasoconstriction, right? Arterial constriction. And then aldosterone increases sodium, and thus it increases the amount of water back into the bloodstream. Okay, so now we've got tighter arterioles and our arteries overall. We've got more water in the blood. So there you go. Our blood pressure will increase. So this is the renin-angiotensin-aldosterone cycle, and it involves renin, which then splits angiotensin into angiotensin 2 using the angiotensin converting enzyme. And then, of course, angiotensin 2 directly stimulates arterial constriction and release of aldosterone. Whole big cycle. All right, and then lastly, we have the ureters. They leave the kidney at the hilus. They have an outer fibrous layer, middle smooth muscle layer, and inner layer lined with transitional epithelium. The transitional epithelium allows the ureters to stretch as urine passes through them on its way down to the urinary bladder. And then there's the smooth muscle layer propels the urine through the ureter by peristaltic contractions. Urinary bladder is lined with transitional epithelium that stretches as the bladder becomes filled with urine, and the wall of the urinary bladder contains smooth muscle bundles. The neck of the bladder extends caudally from the sac into the pelvis canal and joins the urethra. And of course, we'll talk about the urethra in a second because it's quite different depending if it's male or female. Around the neck of the urinary bladder are circular muscles composed of skeletal muscle fibers, which is under, of course, voluntary control. Control of urination, so micturition or uresis is the expulsion of urine from the urinary bladder into the urethra. Urine accumulates until pressure of the filling bladder activates stretch receptors in the bladder wall. A spinal reflex returns a motor impulse to the bladder muscles, causing them to contract. Contraction gives the sensation of having to urinate, and then we have voluntary control of the muscular sphincter around the neck of the bladder, which results in temporary control of urination. So the urethra is the continuation of the neck of the urinary bladder, carries urine from the urinary bladder to the external environment. It's lined with those transitional epithelium, which allows it to expand, and this, of course, is depicting the male. So we've got the two kidneys, the ureters, the bladder, and then the urethra, which comes around in sort of a backward C shape. The female urethra is shorter and straighter than the long curved urethra, and it opens to the ventral portion of the vestibule of the vulva. It's important to note that females are at higher risk of getting bladder infections caused by bacteria entering into the urinary bladder because of their short urethra that is fairly close to the anus. Okay, so because it's short and it's on the caudal aspect of the animal, if they have any sort of diarrhea or if they have challenges with grooming, then bacteria easily enters into the urethra and then into the bladder to cause an infection.
Maybe I was a fool for dreaming that we might just work out fine. And I've been thinking